Alright guys, hello again. In this video I'm going to review transcription super quickly, and then we're going to hit translation, which is the second part of protein synthesis, and then I'm going to very briefly talk about the mutations that can happen on DNA, because we've done that in regular biology and they're pretty straightforward. So let's review transcription. I hope you've watched that video. Remember, there are 3 steps to it in prokaryotic cells: initiation, elongation, termination. And then in eukaryotic cells, they do something a little bit different. It's like an extra safety measure where it's going to add the cap on the front and put the tail on the end, and then take the introns out and leave the exons, which is called modification. So those are the 4, basically, steps of turning DNA into RNA. So today I wanted to focus on translation, which is actually taking the RNA and making it become a protein or an actual functioning gene. Okay, so here's kind of a picture overview of it, and there are some new steps in here, obviously, because when we did this in regular biology, we just talked about the ribosome. We didn't talk about these 3 different channels that are inside there, which I'm going to show you in a minute. But the goal basically is to take this messenger RNA that came from the nucleus and turn it into this functioning protein here, which is a chain of amino acids held together by a peptide bond. Okay, so let's kind of review. Remember, messenger RNA does not have any T's, it has U's. So if I am turning DNA into RNA and I have TAC, my codon is going to be AUG, and then if you remember how to use this particular chart, which of course it would be given to you if you need it on the exam, AUG is actually methionine or start. So it'll be start if you see it at the beginning, it'll be methionine if you find it anywhere else in the chain. So AAA becomes UUU, which is my favorite amino acid, which shows you how nerdy I am. That's phenylalanine. And then ATT would pair with UAA, and UAA is one of our stop or termination codons. So just a quick review on how to do that. Here's a larger image of that chart, but remember, it will be given to you if you need it for some reason on the exam. Okay, so let's talk about messenger RNA and tRNA. So the messenger RNA is the single strand that contains a codon, and remember, the codon is 3 nitrogen bases that code for an amino acid. So see how it's divided into chunks of 3 here. Those are codons. So A, U, C, G, and I'm going to throw you for a loop in a minute. There's actually another one that we didn't learn about in regular biology. Okay, so the tRNA is the one, remember I told you she's kind of like the waitress. She's the one who brings you the right order and puts it in the right place. So your book actually uses 3 models to show you what tRNA looks like. I showed it to you kind of like a T or a coat hanger looking thing, which is the most similar to this here, but you can see there's A's and U's and C's and G's, and then your anticodon will be the one on the very bottom here that pairs up with the codon. Here's a 3-dimensional structure, which I don't care for at all. And then this one that your book uses most frequently, it looks like a little firecracker to me. So the anticodon is here, and it doesn't show you all the other A's and U's and C's and G's that make up this codon. It only shows you the anticodon, but this is the one that your book uses most frequently. So here's the anticodon, and here's where the amino acid would attach to. Okay, so remember, the anticodon pairs with the codon, so that way it knows it's in the correct spot. Now here's something new. And of course we had to make it a little bit more complicated. We actually have 45 different varieties of RNA because of something called Wobble. So whenever there is a U in the 3rd position, which is called the Wobble position, it can actually pair with an A, which it normally does, or a G. So that gives you way many more options instead of just the original 20. Then, take a look at this. There is an actual, another nitrogen base that's called Inascene. An enzyme can change an A to an I, and look what I can do. It is actually very special because it can pair with a U, a G, or an A in the 3rd position only. Now, why it doesn't pair with the C, I can't tell you that. In fact, I didn't even learn about Inascene when I was in biology. Guess it was a new discovery. I have no idea. You know that was a long time ago, by the way. Okay, so it's a two-step process to actually get it to connect to the correct binding site. So what it has to do is remove AMP, and it uses ATP, which I hope that rings a bell to you. Remember, it has to have energy in order for this to happen. So here's kind of an overview picture of it, and I wouldn't worry about that too greatly at this point. Just know it needs energy to create a protein, and you know that because it is actually a you're creating a bigger molecule from smaller ones, which means they're anabolic, right? And they actually require energy to do that. Okay. All right, so here's something new too. I want to show you the sites that are inside the ribosome. Now, there's two ways to look at this. You can actually remember it as the EPA, like the Environmental Protection Agency, because the messenger RNA comes in from this direction, so it will hit the E site first, which makes no sense because that means exit. I like to remember it going this direction because I can remember the word ape. Okay, this one holds the next amino acid. This is where the protein exits. If you notice, there's actually a tunnel to come out the top. And E is for exit. This is where the tRNA exits. So I remember amino acid, protein, exit is how I remember it. Okay, so let's take a look at that. Just like transcription had 3 steps to it, so does translation. Initiation, elongation, and termination, which are very similar. Please don't focus on the energy or any of those things right now. Okay, so the ribosome is actually made of 2 pieces, a larger piece and a smaller piece, and they come together to allow the messenger RNA to ticker tape through here. And then when it's over, they're actually going to disassemble. Okay, so the start codon is here. Here's your small piece. Here's your tRNA, and then there is your start codon, your methionine or your AUG. Okay, so first thing it has to do is recognize the codon. It's got to figure out which amino acid that the gene is looking for. Okay, so your GTP is what provides the energy for this particular step, and you use ATP when you're hooking them together. Okay, so here is the codon of what we need. Notice it is in the A site. This is the one that it's going to add. This tRNA molecule is going to come in here, and notice it has the amino acid attached to it. Okay, so here's your growing protein chain. Here's the next one that needs to be added. So this picture is showing you it's in the site, okay, and then how this particular protein attaches to the very next amino acid that is needed is it actually skips over to the A site. So you can actually see that happening in the very next slide here. See how it went from here in the P site over here to the A site. And GTP is what provided the energy for this to move, and then ATP is what's going to actually combine those together. Okay, notice it asks you the question, where is it located? It moves over here only temporarily to attach to that amino acid. Okay, then for the translocation part, remember translocate means to move. Okay, so this is like one smooth step here, even though it's really, really hard to visualize. So the tRNA that was in here is the one that was previously used, now jumps into the E site, and it's going to exit and go pick up another amino acid. Okay, this tRNA that was originally in this A site and brought this amino acid hops over to the P site. And now, see, this particular protein is actually growing here. Okay, remember, it moves through here in a specific direction. Okay, so we're always going to add amino acids to the 3' end. Okay, so here's a picture of this all together. Your initiation, your elongation, and then your translocation. So that is going to continue until the particular protein is made, right? And it's going to reach a stop codon. Okay, so once it reaches the stop codon, then it knows that the amino acid chain, the protein, is finished. Okay, so here's your protein leaving the ribosome, and then look, your ribosome actually disassembles completely. So then this messenger RNA can actually be recycled. Okay, so another new term here, polyribosomes. You know that just means many ribosomes. So basically all of these ribosomes are making thousands of copies of the same protein. It's like they all have the same instruction, and it moves from one through the next, through the next, through the next, and it creates the same protein. So I would imagine this would happen during a period of rapid growth, or if someone or something was injured and it needed a bunch of the same types of proteins created. That's what polyribosomes are for. Okay, remember that we learned that we have smooth ER and rough ER, and the smooth ER does not have any ribosomes bound to the surface of it, but the rough ER does. Okay, and we've got some other ribosomes that are free and some that are attached. Okay, and this is showing you how this can happen inside of the ER. See how you can have thousands of proteins made at a particular time. Okay, all right, very quickly, let me just go over some mutations. I only have a couple minutes left before I have to end this video. Remember, a mutation is any time you change your DNA. It doesn't matter whether it has an effect or not. Anytime you change your DNA, that is called a mutation. Mutagens are things that cause mutations. So things like chemical radiation, UV radiation, all kinds of things can cause mutations. So a point mutation is when you change one single base pair. So in this example, we went from CTT to CAT. That's a point mutation. This next one is on messenger RNA. GAA became GUA. So instead of creating glutamine, which is for normal hemoglobin, we made valine in this particular spot. So this individual would actually have sickle cell. Sickle cell is a point mutation. Okay, now substitution is just like what we just said, when one nitrogen base becomes something else. So basically three things can happen. Okay, a silent mutation is actually when you change your letter but you get the same amino acid. So remember how serine and valine and a lot of those are in the chart a bunch of times? That's what prevents something from actually happening. So was it mutated? Yes. Did it cause a problem? No. That's a silent mutation. Nonsense mutations is when you actually create a stop somewhere in the middle of your strand instead of at the end. Okay? And then a missense is basically when you change the amino acid that you had intentionally wanted. So like the sickle cell example, when it made a valine instead of a glutamine, that would be a missense. So here's pictures of that. Okay, here are no effect because we still made glycine like we wanted. Okay, here is a missense because we made serine instead of glycine. And then this one is a nonsense because we actually had what we call a premature stop. Okay, and then insertions or deletions. This is when you have an extra nitrogen base either added or deleted. So the reason it is called a frameshift is because the entire DNA strand moves, either one back or one forward. And these are the ones that create the biggest problems, because you're changing way more than one amino acid. So if a frame shift happens really early in the messenger RNA strand, there's going to be a lot of issues. If it happens toward the end, it'll be less so. But a lot of times frame shift ones are the ones that end up causing death. Okay guys, I'm going to end that lesson here because I have like 55 seconds left. So work on the other things that are attached to this, and please, please, please let me know if you have any questions.